All right, so we're going over the phases of cardiac cycle. As you know, the cardiac cycle refers to all the events associated with the blood flow through the heart. Two major things that are happening. One is systole when the heart is contracting, and the other one is diastole when the heart is relaxing. So what we wanted to do is take a look at these two phases. In particular, the three phases of the cardiac cycle is one, a ventricular filling, right? Ventricular filling. As the name of it implies, the ventricles are getting filled with blood, all right? So this is interesting because while the heart is relaxed diastole, all right, the heart is getting filled with blood. Okay, that's what it's saying. And the ventricles are getting filled. Now, the whole heart pressure at this point in time is low, uh, and slowly the blood enters from the atrium into the ventricles. In order for that to take place, obviously the AV valves has to be open, so that way, uh, they could fill it up. At the end of the cycle, atrial systole is occurring. So for those of you that told me you are visual learners, I'm trying to give you guys the idea visually so you can understand what's going on. So pretend that this is the heart atrium, all right? And you can maybe have the ventricles at the bottom, all right? So the atrium, here is the AV valves, here is the aortic arch, here is the uh, right AV valve, and I don't know if it's going to take a little bit longer, but that would be the pulmonary vessel. And as you know, we have a semi-lunar valve right here, aortic semi-lunar valve, and the pulmonary semi-lunar valve. This goes towards the lungs. Previous lecture, we mentioned that, huh? All right, so this is the right atrium, and this is the right ventricles. Now, ventricles is better to withdraw it with this, so we could have the left ventricle that was have a thicker myocardium, if you remember, and you have the right left atrium. All right. So at this point in time, blood coming in from the lungs, it gets oxygenated and it's coming back to the pulmonary vein, if you remember. So you're receiving the blood on the atrium. That's what atrium do. All right. And from superior vena cava and inferior vena cava, they're both pretty much running into the right atrium. So superior and inferior vena cava, okay? And the blood is coming. These are the deoxygenated blood. On the other side is oxygenated blood. So if the blood is coming in and these valves are open, then it goes right into the ventricles. The ventricles, pretend this is the blood, is getting filled with blood. All right, it makes sense for the AV valve to be open at this point in time. That's the AV valve. This is the right AV valve. They're both open currently. At this point in time, we are in the middle of diastole. That means the heart is relaxed. It's not moving. It's not contracting. All right. But remember, early on in the ECG, we discussed the P wave. And the P wave where you had the SA node, and we mentioned that the SA node is basically the nodule cells that initiate their own action potential, and then is spreading throughout the atrium. So the atrium contracts first. So as the SA node, pretend is here, and relaying the electrical impulses, that's the AV valve, AV bundle, SA node, and AV node. and a bundle of his and all that. So basically, what is happening, the atrium is going to contract first. Again, go back and review the earlier lecture I gave you, but from the uh, autorhythmic cells, the signal is going to go to the contractile cells. So as a result of end of this cycle, we are still in ventricular filling. The ventricles are getting filled with blood. All right, the blood is returning to the atrium. The atrium then need to contract. So this arrow showing you the atrium is contracting. We are probably in the middle of the P and going towards the end of it. At this point in time, again, I'm going to show you in the dotted line. Pretend that the atrium is now smaller because they contracted. Obviously, what is going to do? What is going to do is going to raise the pressure in the atrium. Okay, the pressure is going to go higher. All right. Imagine that you have a balloon and you're squeezing it, and inside of the balloon you have water. All the water is going to come out because you increase the pressure as you squeeze the balloon. Same thing in here. The heart is kind of like a balloon in this case, and the blood inside of the atrium is kind of like a water inside of the balloon. So now what? Well, the pressure always goes from higher to the lower. Inside the ventricle is lower. So now it's forcing because they cannot go backwards. So they're going only this way. Now they're getting filled even more with blood. All right. So at this particular case, these are the SL valve. There are two of them, one here and one there. Huh? The SL valve is closed. So during this event, we could summarize it as AV valves are open, SL valves are closed, pressure in the atrium increase, all right? And the volume is decreasing in the atrium. Why? I just told you. Because it's pushing all the blood or majority of it towards the ventricle. At this point in time, volume is increasing in ventricle because the ventricular filling, obviously the volume is going up, all right? But the pressure is low in the ventricles, all right? The pressure is low because it didn't contract yet, all right? Remember, the electrical impulses, they need to go first towards the myocardium, then it's going to come over here, and then it's going to go from the Purkinje fiber and it's going to go to the myometrium. So next up is the contraction of the ventricle. But before we see that, we need to go back and take a look at what is the next step. So this one is done. We are done with the ventricular filling. Next up, you have ventricular systole. Ventricular systole when the heart is contracted, obviously. At this point in time, we are, in terms of the ECG, we are done with the P wave. So imagine that the atrium or depolarization is already occurred. Now we're going towards the repolarization of the atrium and also depolarization of the ventricle. Do you remember on the ECG, at which point in time depolarization of the ventricle was occurring and and also repolarization of the atrium? Do you guys remember? If you want, you could pause the video and see if you could remember. This was the P wave. What was after that? QRS. And again, this is the moment in which the depolarization of the atrium beginning, right around here on the Q area. All right. But we don't see the contraction until the R area. Right here is beginning of the contraction. Because electrical impulses, they need to travel and go to the myocardium, that we are going from the autorhythmic cells to contractile cells, and that takes some time. And then they need to go through the intercalated disc, the gap junction, and they need to spread. So then the contraction starts, which again, the spreading part of it will take a little bit of time, and that's why it's not at the beginning. Regardless, this, you need to know the corresponding ECG. How is it related and why? It's better to know it rather than memorize it, or you have to look it up to get the points and so on. You need to learn this. It's interesting once you understand the concept. Okay, so atria is relaxing. Makes sense because repolarization of the atria. So right after that is relaxation, obviously. Rising ventricular pressure result in closing of the AV valve. All right. So what this means is we got to go back, and maybe I can just show you right over here. All right. So again, here's the heart. The heart was filled with blood already, came already in here. So here's our ventricles. So it's rising. Okay. Now it's contracting. Remember, by this time we are at the QRS. So this is depolarization. Depolarization followed by contraction. So we mentioned this already. As soon as they reach up here, boom, they're going to close this valve. No more. The valve always prevent backflow unless you have a defective valve. So what that means, they cannot go back that way. I mentioned this earlier. It is kind of like, you're putting air in your tire, all right? It won't come out anymore because of that valve. All right, now, what happens next? Well, at this point in time, there is a rise in pressure. Did the volume change? No, it didn't change because it didn't exit the heart yet. We're still inside of the ventricle, but there is less, there is more pressure because again, you think about that you're squeezing the balloon again, right? So the pressure is going higher at this point in time. Where? In the ventricle. What about in the atrium? Atrium has low pressure at this time because they are relaxing. They are relaxing, all right? What about the volume? Well, right now, not a whole lot of changes occurring in the volume, right? So remember, you already contract and all of the blood already went in here. Now they are relaxing. Yes, there will be some blood that is returning again very soon, but we are going over the ventricles right now. Okay, so that's what happens. All right, what else? Let's go take a look. So pressure rising, that's what it means. AV valve is closed. I just explained to you. Now there are two sub phases. One is called 2A. We're going to call this 2A, and then we're going to call this 2B. One is isovolumetric contraction. Iso means the same. Volumetric, volume. Contraction is contracting. The ventricles are contracting. But at this point in time, the volume still is the same. All right. At what point in time there is an isovolumetric contraction? Let's go back. All right. Remember, this was the pulmonary artery, and this one was the aortic arch. And pretend that there is a SL valve here that was closed. But let me see if I can put in the blood so you could see it. As more and more contraction is occurring, they can't go through the valve, so the blood has no way but to go up here, up here, up here. So remember, this valve was previously closed. This one, maybe it's better to show it in blue color to demonstrate that it's deoxygenated blood. And let's just put in the valve that are closed at this point in time. All right, so up to this point, see what is happening. What is happening, you have less room now, all right, in here because it contracted, all right, in here because it's contracted, pretend that the heart muscle is contracted, all right, so all of these is now blue color, huh? These are blue color, all right, all the way up to here. On the other hand, The other side that is oxygenated blood, again, right here, coming all the way up here. So imagine that you're squeezing your balloon, and inside of your balloon is water, right? But you're not letting the water go. The water keeps going all the way to the top. I usually do a demonstration in the class. You have a water bottle halfway filled with water. All right, if you squeeze the water bottle, the plastic water bottle, you notice that the water is going to come all the way to the tip. All right, don't let any water get out. Now let go. What will happen? Did the volume change? No. Why not? Because it's the same amount of water inside of your plastic bottle. Didn't change because you didn't squeeze it hard enough for the water to come out. At this point in time, this is very much similar to what happened. In other words, they haven't left the heart yet. So this is called iso, means the same. Volumetric, volume is the same. Isovolumetric contraction. The heart is contracting. But the heart does not, contraction does not stop in here. Remember, we started at R. We are going towards the T wave now. All the way to the middle of the T is the contraction. Now, the second part of 2B, that is called the ventricular ejection. Ejection, when something is ejecting, right? Ventricle. What is it ejecting? Well, this is going to be ventricular ejection. Now is exiting the heart. The blood is exiting the heart, and because inside the pressure what keeps going higher and higher and higher, is enough to open up the SL valve. So during this phase, SL valves are open. During the 2A phase, the SL valves were closed. AV valves were closed as well. So we could go back and kind of like put in all valves closed, okay? Because the AV valves were already closed, and the blood came in all the way to the SL valve, but the contraction or the amount of pressure was not high enough to open them yet. When you say ventricular ejection, that means what? That means AV valve are still closed, but the SL valves are open. It's enough pressure exerted to open these valves. But these valves are still closed. This was the AV. AV means atrioventricular. AV between the atrium and the ventricle. Okay? So this is known as ventricular ejection now. So, couple of things. All right? The 2A, when we said that there's an isovolumetric contraction and the blood reaches this AV valve and the AV valve closed, that's where you hear the very first sound of the heart. Earlier, I gave you guys an idea about, the sound of the heart, if you remember. And we mentioned they have two sound of lub dub. Lub and then dub. The lub sound is due to the closure of the AV valves. All right. So again, learn why. I just mentioned to you, the valves are closing, making noises. Lub. Okay. And we mentioned this is during the 2A now. Right? Now, the dub sound is actually related to the closure of the SL valves, but we haven't got there yet. Why not? Because right now they're open. We are barely doing the ejection, ventricular ejection. So yes, we're not there yet. All right. So in terms of the volume, and the pressure, isovolumetric contraction, yes, the pressure is high in ventricle. Instead of memorizing, just think of it. It's really easy to figure this on out. Pressure is high. How about the volume? Volume is the same. I will use this like no change volume. Isovolumetric. That means volume doesn't change. The volume is the same. All right. That's what it is. Okay. Pressure keeps on increasing. The volume is staying the same. Where? In the ventricles. Right now, we're just talking about in the ventricle. All right. How about the ventricular ejection? What do you think the pressure is? Pressure is still high. What do you think the volume is? If you want, you can pause the video and guess or think of it. Give you a moment. Pause the video. Is the volume high, low, or no change? Because remember, the volume went out of the ventricle. We are talking about the volume inside of the ventricle. So if they are ejecting blood from the ventricle, the volume is going down. So in this case, the volume and the pressure actually have the opposite relationship with one another. The more pressure, the exiting the blood from the heart, causing the volume to go down. All right, so we are done with the second phase. The third phase happened to be the isovolumetric relaxation. What is happening during the isovolumetric relaxation? Again, iso means the same. Volume doesn't change. This is also early diastole. That means the heart is beginning to relax. So at this point in time, the ventricles are relaxing. All right. They haven't got it filled up yet. So what happens to the volume? Well, they haven't filled up. Otherwise, it would have been called ventricular filling. So in this case, the volume does not change. Iso volume, that means no volume changing. All right. There is a little bit of backflow is occurring. All right. But from the aorta and the pulmonary trunk, and that causes the seminal valve to close. Let's see if we can visualize this a little bit. All right. So now, as you eject it, what happens is a little bit of backflow is coming. They're coming back in here because now you have to erase all these arrow. You have to erase it now, okay? Because there's no more contraction. Now you have to maybe put in the arrow the opposite way. And this arrow is gone too, because we already went over that, right? So what happens is now we have to put the arrow this way because they are relaxing, all right? So there is a little bit of backflow is occurring, but these valves are one way only. So as the backflow occurs, boom, it closes the SL valve. All right. Remember, at this point in time, AV valve is still closed. So these two parts is not that far apart from one another. All right. So at this point in time, all valves again is closed. This is what happens in isovolumetric relaxation. All valve, SL and AV valves are closed. What happened to the volume? Unchanged. You might say, wait a minute, I thought they had low volume. Yes, but no longer is getting lower. So it's unchanged from the point that you stopped. From the ventricular ejection, whatever that amount was at the end of the ejection, it still is that same amount, right? What happens is a backflow is occurring from the pulmonary trunk, which is here, and the aorta, which is this one. And then again, boom, it's closing the SL valve in this case. And that's what it's trying to tell you right over here. So volume is unchanged, backflow is occurring. And this is sometimes referred to as decrotic notch, a brief rise in aortic pressure causing by backflow of the blood rebounding off the seminal valve. At this point in time, you hear the second sound. This is when the SL valves are closing. Where are we that on the ECG? Okay, the first sound was a lop sound. We were right at the beginning of the Q, where the systolic pressure was again started, a little bit after that. But right now, where are we that in terms of the ECG? All right. Well, at this point in time, several other things are happening. Before I tell you, let me go ahead and tell you. After the QRS was a T wave. The T wave representing depolarization of the ventricles. So when they are depolarized, very soon after, they're going to be relaxed. All right. Diastole is going to occur in the middle of the T. That's what it's going to show. All right. Why not at the beginning of the T? Because it takes time for repolarization to take place and the spread, and the potassium ion leaves, all that stuff that we discuss about the action potential. You have to picture every single thing that I told you earlier to get a great understanding of what is happening. Okay. Now, let's go ahead and take a look at the dub sound. The dub sound, or the second sound, is occurring right at the vicinity of the T wave, because as they are relaxing, a backflow is occurring, and that's where the SL valve is closing. So it's got to be right after the middle of the T, all right? And you wanted to understand these parts. So the next slide demonstrates everything you need to know, but you really have to understand this in a great detail. On top, you have the ECG. Again, pay attention as the color changes. It tells you, for example, right over here, it says ventricular filling. So these are all in this blue color right here. Ventricular systole, 2A and 2B. It happened to be kind of like in this pinkish area. And finally, the isovolumetric contraction happened to be after that, followed by ventricular filling. So we're going from the beginning again. All right. So again, at the isovolumetric relaxation, this is where the both of these valves are closed. All right. Again, we'll go back to the ventricular filling. So this is pretty much at the beginning of the ventricular filling. If we go ahead and take a look at the lub dub sound, again, you could see the first sound happens right after the R, where the contraction is occurring. Eventually, the blood reaching the AV valves are closing. And then the second sound, again, is right after the middle of the T. And again, if you have to understand the systole, this area would be the systole. And over here, all the way to the next R, that would be the diastole. It's premature in here, but if you wanted to draw that, that would be all the way to here. That would be the diastole. What can we see from here? Look, the relaxation is a lot longer than the contraction. It contracts much lesser time, approximately 1/3 lesser time. You know, this is like 2/3, right? Diastole is a little bit further, right? In other words, a heart takes longer to get filled. Contraction part of it is quicker. The volume and the pressure is telling you, again, you could look at that, all right? And finally, which valves are open and closed, and what are the things that are happening. If you pinpoint and go over these, you could see what is happening on each one. I think I mentioned that already with my own drawing, but it would be a good idea for you to go through them, make sure you are comfortable, and you know exactly what is happening. All right. Now, the only thing I wanted to stress is probably go a little bit over the stroke volume, because I wanted to talk a little bit about the stroke volume. All right. If you can take a look at this idea, the stroke volume is basically the amount of blood pumped out of the heart by a ventricle with each beat. So again, if you have your water bottle next to you and it's filled halfway, all right, let's just say it's certain amount of volume inside of your water bottle, and you squeezed it out, all right? So you squeeze with your fingers on the surface around it, and let's say that you had like 50 milliliter inside. half of it came out, 25 milliliter came out. What did you start off with? 50 milliliter. What did you end up with? 25 milliliter. What is the amount that came out? Another 25 milliliter. What if you started with 100 and when you squeezed, let's say 60 milliliter came out? What do you have remaining? 40. Well, that 60 milliliter that came out, it's kind of like if you imagine that your water bottle is one ventricle, all right, and that amount came out, all right, that is called the stroke volume. In other words, when you're squeezing the ventricle, what is the amount of blood coming out during the ventricular ejection, all right? So this is called end diastolic volume. This is what you start off with, say 100 milliliter in your water bottle. This is end systolic volume at the end. So you squeeze, you squeeze, you squeeze. At the end, what is the amount left? Let's say 40 milliliter was left. The stroke volume is the difference between the two. So in this case, in our example, would be 60 milliliter, right? So we need to talk a little bit about a stroke volume because it's such an important concept. Cardiac output is equals to heart rate multiplied by a stroke volume. Heart rate, we already know. You can actually take your pulse for the heart rate. How many times your heart beats per minute. You could also look at your ECG to get the heart rate. Do a calculation I taught you last time, all right? So you could figure out your heart rate several different ways. One is pulse rate, same thing as heart rate. Because when your heart beat, contract and relax, you know, the blood vessels expands and go back to the original form. And that's how you pick it up. If you put and palpate your blood vessels of radial artery or carotid artery by your neck, all right, you could figure those out, all right? The stroke volume, again, I just told you, the difference between the end diastolic volume and end systolic volume is the stroke volume, all right? So these are the amount of blood pumped by the ventricle with each beat. All right. So here's a good example of normal cardiac output. So you can see heart rate multiplied by stroke volume. Let's say a person's heart rate is 70 beats per minute, and the stroke volume is 70 mil per beat. You multiply by together, you get 5.25 liter per minute. How do we regulate the stroke volume? All right. So again, we need to have an understanding. If I give you a calculation, I'm probably going to give you an end diastolic and end systolic volume, and easily you could figure out the stroke volume, obviously, right? So you could calculate that a stroke volume as well. The heart rate, you could take the pulse and figure that one as well. All right. So what are the factors affecting the stroke volume? Obviously, we need to regulate the stroke volume as well, all right? At some point in time, we need more cardiac output. In other words, what is the amount of blood that is required by your tissues? It's cardiac output, all right? If you're exercising, your heart needs to beat faster, okay? And if you're at rest, your heart has to beat slower, for example. So at different times, we need more or less cardiac output. When you're exercising, your heart rate goes up. That makes sense because you wanted to increase the cardiac output. Even if you don't know what happens in the stroke volume, just by taking the heart rate up, assume that we don't have the value in here, cardiac output most likely is going to go up as well. But we want to know what happens to stroke volume at different instances. But before we do that, during exercise, during the sympathetic activity, parasympathetic activity, blood loss, during dehydration, during the availability of higher amount of calcium, all of those things can happen. But we wanted to talk about the factors affecting the stroke volume. One is called preload, the other one is contractility, and the third one is afterload. All right. So preload, amount of ventricles are stretched by contained blood. Just like a balloon, if you fill it up with water, it expands a little bit. Now you can have more water inside your balloon because it stretches. This is referred to as preload. All right. uh, Due to the way that the actin and myosin position themselves and so on, they could stretch, and the amount of blood that is returning to the heart might vary as well. All right, so that is called preload. In other words, preload usually referred to the amount of blood that is coming back towards the heart. If it's a lot is coming before they load up, before they contract, that's called preload. Contractility refers to the force that is required for myocardium to contract. And finally, you have the afterload. What happens after that? There is a back pressure exerted by the blood in the large arteries, such as aorta, as the blood is leaving the heart. And that is called afterload. So, uh, let's go ahead and take a look at these one by one, see if we can make a sense of it, and then we can have a understanding of what's going on. Okay. Sierra and Rob, what are you guys doing? Huh? Make sure your light is on. You have good flow of light. Rod, what are you doing? Good job. Good job. Francesco, can you hear me? Put on your speaker because I cannot hear you. Francesco. Okay, I unmute you. Okay, hold on a second. Okay, do you remember the questions on your exam?
The 6 questions.
The numbers. You took a screenshot, right?
Oh yeah, I took a screenshot.
Okay. Tell me which question was. I think one of them was biceps brachii. You answered it correctly, so that was done. Another one that wasn't loading was number 18. It was the inferior gluteal nerve. You answered correctly, so that is out of the way. There is a next one that was the deltoid. You answered it correctly, so that was out of the way. So
I think it was question 17 until 21. Oh no, 22.
okay, so question 17, you got it right. I just told you that.
Oh yeah.
And what else? 22?
Yeah, so yes, 22.
So 22, it was a tricep, and you already answered it as radial nerve. So you got those two already. So it didn't affect you because you were answering it based on the written words.
Oh, okay.
You know what I mean?
Yeah.
So uh, is there anything else I could do for you?
No, I think I'm good. Yeah.
Okay. I haven't looked at the overall. You know, you might be able to get partial credit here and there. I don't know. I have to look at it.
Okay.
You know what I mean? But I'm not finishing up with your exams, but it looks like those that you had problem with, you went based on the wording, correct?
Yes.
Okay. Again, I will look at all your questions. If there is anything like, you know, partial credit or something I could help you with, I will do that. All right? That's going to happen at the end. So we're good to go.
Okay. Thank you.
All right, you're welcome.